Whoa, would you look at the time? It's time to learn about molecular genetics. Okay, so first, what are molecular genetics? In this video, we'll mostly be covering Mendel's rules for inheritance, as well as more recent discoveries such as complex inheritance patterns and human pedigree. So Gregor Mendel was a German monk who in the 1850s worked with pea plants using the scientific method to determine the two laws of inheritance, which is why today he's known as the father of modern genetics. He also discovered the existence of alleles, which are just alternative versions of genes, which are coded by specific DNA sequences on a specific chromosome, when are inherited twice per organism, once per parent. He found out that there are two kinds of alleles, dominant and recessive. Dominant ones are expressed through physical traits, but the recessive ones, while they are passed down, are not actually physically expressed. The laws of inheritance are the laws of segregation and the law of independent assortment. We'll start with the law of segregation, which states that alleles separate during meiosis, meaning that half the gametes end up with the dominant allele and the half end up with the recessive, and this corresponds with the separation of homologous chromosomes during meiosis. Gregor Mendel described phenotypes and genotypes, phenotypes being the physical characteristics of an organism, like its height, its weight, and its color, and the genotype are its genetic makeup. We can determine the offspring's genetic makeup using a Punnett square by putting one parent's genetic makeup over the top and the other down the side, and filling in the combinations, we can determine the genetic makeup of the gametes.
Hey guys, it's your boy Dennis Liu coming at you with another AP biology review video. Well, in this case, it's the second half of the molecular genetics video. In this part of the video, I'll be talking about complex inheritance patterns, the law of independent assortment, and finally, human patterns of inheritance, which includes pedigree testing. So let's get right into it.
First up is the law of independent assortment. So what this means is basically each pair of alleles segregates independently of each other pair of alleles during gamete formation. So what this looks like in practical terms is that it just your pairings do not matter, right? So in a dependent scenario, your dominant alleles would align with dominant alleles and recessive ones would align with recessive ones. However, in the independent scenario, this is not the case. So just a quick rundown of what Gregor Mendel did is that he started off with homozygous yellow and round peas and homozygous green and wrinkled peas, and then he crossbred them. So then this yielded all yellow round peas, but this is heterozygous. So then if you were to breed them again, you would have two possible scenarios yielding two different ratios with phenotypes. So what Gregor Mendel did was that he actually experimented with this and he compared his actual real life results with the theoretical ratios, and he found that the independent scenario was the one that matched up with reality. Okay, so quickly running through complex inheritance patterns. One example would be incomplete dominance, where hybrids have phenotypes in between the two parental varieties. For example, observe the top example. So if you have a red flower and you breed it with a white flower, the result would be a pink flower. This is just a mixing. And another example is codominance, where two alleles affect the phenotype in separate, distinguishable ways. So in my example, you have a white cow, made it with a brown cow, you get a white cow with brown splotches. All right. And in addition, there's pleiotropy, which means that one gene has multiple phenotypic effects. Epistasis, where the phenotypic expression of a gene at one locus alters the effect of a gene at a second locus. So here's a really quick rundown of human patterns of inheritance and pedigrees. So pedigrees are diagrams illustrating family relationships and the genotypic/phenotypic traits of each individual. So shown here is an extremely simple pedigree displaying a small family with dwarfism running through the family. So right here, you have a male with dwarfism and a female who does not. So they come together, have children, and they have a daughter who has dwarfism and a daughter who does not. So just through these pedigrees, you can really start to see how certain genetic disorders behave or are transmitted or expressed, and these are used typically to just learn more about them. And so dwarfism isn't really a good example, but for something like a sickle cell anemia, even though this girl might not express sickle cell anemia, she might be a carrier, which are people who are phenotypically normal in regard to the disorder, but they can transmit the recessive allele to their offspring.